class on yesterday. So yesterday morning I need to attend a meeting, so I cannot do the class. So your class representative, I think he already discussed with all of you. So you choose this time, from 1 to 2 p.m. Okay, so this time, in this session, let's listen to the presentation, from the 3 students. So are you ready? So the first topic is about the structure of ubiquitin and proteasome. So who's going to present this one?
Me, doctor.
Okay, wait. Okay, hold on. Okay, siapa? Ain ke siapa? What's your name?
Ain, doctor.
Ain, okay, Ain. Okay, Siti Nurhaliza. I tanda your name. So you did one presentation. Ain, Siti Nurhaliza. Ain, you can share your screen now.
Okay. Doctor, can you see the screen?
Yep. Okay, let's listen to Ain. Okay. And
Doctor.
let's ready with
Wait, doctor.
Yep. Mm-mm.
Can doctor listen to me?
Yes.
It's my okay.
Okay, Ain, whenever you're ready.
Okay, I will start now. Assalamualaikum and good morning. So I will talk some stuff about ubiquitin and proteasome today. So first of all, what is ubiquitin? So ubiquitin is a small basic protein, and as you can see from the right side of the picture, it's about eukaryotic cells. So ubiquitin is widely distributed in eukaryotic cells. At the same time, they are also highly conserved among eukaryotes. So you already know ubiquitin. How do they look like? So the structure of ubiquitin is that at carboxyl terminal, the glycine residues attach to the lysine residues at the target protein. So this is how they look like. So when you know about ubiquitin, what their function is? So what do they do? So ubiquitin, as you can see from in front of you, ubiquitin will covalently attach to the target protein. So this protein will be marked for breakdown. So all these for all this process, we call them as ubiquitinations. So ubiquitination is some kind of tagging system. Who's tagging whose? Ubiquitin will tagging the target protein. So when we talk about ubiquitination, they have many kind of form or tagging. So you can see mono, they can also be in multi, they can also be in change, they also can be in mix or also be in branched. So you already know what do they do. And along the way, there are 3 types of ubiquitin that you will be very familiar with. So these three types of ubiquitin is three ubiquitin enzyme. So the first one is E1, ubiquitin activating protein. So what do? They catalyze the first step in the ubiquitination reaction. So an easy way to see is that E1 will activate the ubiquitin. Next one is E2, ubiquitin conjugating enzyme. So the activated ubiquitin from E1 will be transferred onto E2. At the same time, E2 will also recognize the targeted protein. And the last one is E3, ubiquitin protein ligase. So E3, they mediate the transfer of ubiquitin, activated ubiquitin from E2, ubiquitin conjugating enzyme, to specific substrate protein. So if you're a little bit confused of what I'm trying to say is that ubiquitination have three process: activation, conjugation, ligation. From activation, you can see E1, they will activate the ubiquitin. Then the next one is conjugation. So E2, the activated ubiquitin from E1 will be transferred into E2, and at the same time, E2 will also recognize the target protein. So at the ligation, E3, they will transfer the activated ubiquitin from E2 to the target proteins. So what happened to target protein? They become polyubiquitinated proteins. They can be in many kind of form that I said before. And then you can see from the next one is proteasome. So what is proteasome? Proteasome is a large protease complex, 26S. They specifically degrade ubiquitinated proteins. And they have two major domains, 20S core and 19S caps. So what is their structure? So at the blue one, 90S domain, something happens such as ubiquitin recognition. At the same time, at the yellow part, 20S domain, proteolysis occur. So what do they do actually, proteasome? So at 90S domain, ubiquitin target protein will be recognized first. So you have seen at the ubiquitin process just now. And then when they enter the 20S domains, the protein will be unfolded. So at 20S domain, the protein will be degraded into peptides. Ubiquitin at the same time, they will not be degraded, but they will be released and recycled again. So as you can see from the picture, this protein, this ubiquitinated protein, it is like the ubiquitin, and this below is the peptides. So so about ubiquitin and proteasome, they actually involved in the cellular protein degradation. So cellular protein degradation, they have two pathways: the ubiquitin proteasome pathway and lysosomal pathway. But I will only talk about ubiquitin and proteasome pathway because they involve, of course, ubiquitin and proteasome. So ubiquitin proteasome pathway, their function is that they degrade abnormal proteins. They at the same and they also degrade short-life cytosolic protein. They also dependent on ATP and also operating in cytosol. And two type proteins that involve is ubiquitin and proteasome. That's all from me. Thank you. Done, doctor.
Okay. All right. Thank you, Ain. So any question to Ain from the class? Anything that you want to ask regarding the topic?
I think no, doctor.
Hmm?
I think no, doctor.
No. No question. Semua faham. Okay, I would like to ask one question. So you mentioned that the ubiquitin, it will be recycled, right? It will be reused for the next cycle. And is it, in case for the ubiquitin, does it have half-life as well, like the other enzyme or protein? Because you know that protein and enzyme, they have their own half-life. So what about ubiquitin?
Is this question for me, doctor?
Okay, for all of you. Or I can choose anyone. I know everybody is Googling now. Okay, Razik. Are you there, Razik? Okay, what is your answer for my question?
Um, tengah cari.
Kena cari. Lamanya. Okay, what about others? Dan Shin Er? Are you there?
I think yes.
Uh-huh.
I think it's 28 to 32, 31 hour.
You Google lah. Okay. So why do you think that Biquitin also have half-life? Shin Er?
I'm not sure.
Okay. All right. So Dila said because it's also a protein. Yes, it's also a protein. So it's also have, of course, it has such half-life like the other protein or enzyme. Okay. So basically, when you said that protein will be recycled, will be used for the next cycle. So meaning that as long as it's still, within the period of activation, okay, so it keeps running, doing all those staging work. But then at the end, it still also have this expiry date, right? Okay, so all right, so thank you, Ain and all. So the next presenter. So the next presenter is going to present on the disease related to optimality of protein turnover. Yeah, quite interesting. So siapa dah presenter ya for this topic? I think I dah lupa dah siapa ni. Who is that? Let's please introduce yourself. Razik ah? Razik. Okay. All right. Wow. Okay. Oh, nice slide. All right, so Razik, whenever you're ready, so the floor is yours.
Right. Good afternoon, everyone. So I will present about the disease related to abnormality of protein turnover. So protein turnover is the process of synthesis and breakdown of protein, and it is a dynamic process that needs to be regulated well. If this process is not regulated well, it will cause the abnormality of cell behavior. So one of the disease that is related to protein turnover is phenylketonuria, also known as PKU. So PKU is inherited disorder where people with PKU cannot process phenylalanine from their dietary taken or protein from the dietary protein, and they lack of uh, phenylalanine hydrolase enzyme that convert phenylalanine to tyrosine. So the high amount of phenylalanine in their body will cause harmful effect to their body, and this is where they will disrupt the cell behavior. So this is the reaction where the phenylalanine from dietary protein and from tissue protein cannot be converted into tyrosine as the lack of phenylalanine hydrolase enzyme. So this is how people with PKU not be treated looks like. And yeah, PKU is a toxic level of phenylalanine, common protein amino acid that can be found in any of our food, will cause harmful effect to our body. PKU can cause mental retardation, convulsion, behavior problems, skin rash, and musty body odor. So people with PKU cannot take phenylalanine too much from the diet. So that means no meat, no dairy products, no dry beans, no nuts, and no eggs. And they also need to take cereal, fruits, and vegetables in moderation. So this is the example of food that contain high phenylalanine, fish, milk, meat, and so. And this is what people with PKU should take. Most vegetable, fruits, sugars, low protein food, and special bread, cookies, crackers. So the treatment for people that have PKU is to control their diet by taking low phenylalanine foods. These are the examples. And also there are pharmacologic treatment. And as for newborn, PKU disorder can be detected at birth before any symptoms show up by screen check the newborn by test the elevated level of phenylpyruvic acid in their blood. So that's all for the disease related to protein turnover. Dah, doktor.
Okay, thank you, Razik. Okay, any questions, Razik? Okay, what do you think about the choice of the disease that related to protein turnover that Razik presented just now? Come on, class. Do you think that the disease is related directly to protein turnover? Not sure. Okay, Razik. So you choose phenylketonuria, right? PKU as the topic is the disease that related to abnormalities for protein turnover. Okay. So what makes you think that protein turnover related to the pathway? Pathway for phenylalanine degradation to tyrosine.
So, alright, so people with PKU, they lack of phenylalanine hydroxylase. So the phenylalanine they taken from dietary protein or even from tissue protein, they cannot be degraded into tyrosine. And this tyrosine will be will undergo oxidation and protein synthesis, and that's all. So when phenylalanine cannot be converted into tyrosine, they will people with PKU have high amount of phenylalanine that has harmful effect to our body. For example, people with PKU will have urine seizure and also hypopigmentation.
Okay. So yeah, so thank you, Razik, for your presentation. Okay, so the next presenter, we have the topic of autophagy and fasting. Okay, so the next one is Yalini. Okay, so Yalini, the floor is yours.
Can you hear me? Okay. Hi everyone. Now I'm going to present about autophagy and fasting. Okay. So as for the first slide, what is autophagy? Okay. So autophagy is actually a natural cellular mechanism by which the cells in our body degrade unnecessary and damaged components within the cells. The process of autophagy actually helps maintain normal functioning, which is homeostasis in the cell. Autophagy is the body's way of actually cleaning out damaged cells in order to regenerate newer and healthier cells. So in literal, actually, auto means self and phagy means eat. So the meaning is actually self-eating, which is also referred to as self-devouring. While actually that might sound like something you never want to happen to your body, it's actually very beneficial to your overall health. Why? It's because autophagy is an evolutionary self-preservation mechanism through which the body can remove the dysfunctional cells and recycle parts of them towards the cellular repair and also cleaning. And autophagy forms a part of metabolic process which helps the cells to convert food into a form of energy that cells can use to grow and also divide. And metabolism balances between two opposing activities, which is anabolism and also catabolism. So the next is, what is autophagy in fasting? Next slide. So intermittent fasting is a possible way to actually induce autophagy. Under normal conditions, when the cells have sufficient nutrition, autophagy degrades damaged components in the cell. When fasting starves the cells, autophagy helps to digest energy for survival. And also research has indicates that the intermittent fasting, calorie restriction, and also ketosis can all trigger autophagy. Depending on the individual's metabolism, significant autophagy may actually take 2 to 4 days of fasting in humans. Autophagy is believed to begin when glucose and insulin level drops considerably. Animal studies have actually shown evidence of autophagy after 24 hours of fasting. And also some studies have also detected that autophagy in human cultured neutrophils, um, which is the most abundant type of immune cells in the body after 24 hours. But there's no conclusive studies on humans that indicates an optimal period of fasting to achieve autophagy. So that's all from me. Thank you.
Okay, thank you, Yalini, for your presentation.
Dr. Angeline, Yani helped me to present.
Okay. Angeline, sorry.
Yeah.
Okay. Yeah. Okay. So, Angeline, so how different is the process or the function of autophagy, okay, during the normal condition and also during the intermittent fasting? Do you know? Do you happen to read it anywhere about it?
Sorry, doctor, can you repeat the question? My line is breaking actually. Sorry.
Okay, so we know that autophagy can eat up the cells, okay, during the process. Okay, but then what is there any difference between the process of autophagy during the normal condition, which is the normal one, and the one the process of autophagy during the intermittent fasting? Is there any difference?
Not so sure, doctor. I'm not so sure about it.
Okay. So any guess?
Doctor, is it autophagy generate new cell while protein turnover generate new protein?
Sorry, again.
Is it autophagy generate new cell while protein turnover generate new protein?
Generate new cells. Can you elaborate more, Nadira?
No, autophagy kills cells while protein turnover generate new protein. Is it like that, doctor?
Okay. Most probably. I'm also not sure. But I'm asking you about the, something that you can relate to the process of autophagy and the intermittent fasting. And because just now, Angeline presented about the, during the intermittent fasting, the autophagy is the process is really efficient, right? Compared to what she presented just now. So that's so my questions coming asking about the difference between the efficiency between this autophagy process during the normal and during the intermittent fasting. So you said just now, Nadira, you said the the autophagy eat up the cells.
Yeah, it clean out damaged
Yeah.
cells.
Damaged cells. So you mean probably more efficiently, right?
Yes.
Yeah. Okay. So fasting trigger autophagy. Yes. Yeah. Okay. During hunger, body will trigger the autophagy to get extra energy. Yes. Yes, that's right. Okay. So so we have covered on 3 small topics today. Okay, so thank you, Adeline, for your presentation. Okay, so So today we covered on the ubiquitin, right? Ubiquitin and also protein turnover and also the autophagy. Okay, so these three topics, yeah. Hopefully these three topics helps you to do your revision for your test one. Yeah, somehow you can refresh whatever the ideas or some facts about these topics. Okay, I'm going to share slides. Just hold on. Okay, can you see the slides? Yes, yeah. Okay. So I'm going to continue a little bit on this topic. Okay, for today. Okay, for today, because on the next week, we're going to have class as usual, and on Tuesday we have holiday, it's public holiday, and then on Wednesday we have 1 hour class, and then in the lab session you're going to have your test run. Okay, and so we have about 25 minutes or 20 minutes. So I'm going to continue a little bit on this topic, amino acid anabolisms. So previously we have learned on the amino acid catabolism. So how the amino acid is degraded into a simpler molecule or simpler elements, yeah, such as the as we have discussed before, how the certain amino acids like glutamate, serine, glycines, and all that, how it will be degraded into the pyruvate and the other protein and the other amino acids. This is degraded into other intermediates that play a role in the TCA cycle and other cycle as well. And we also discussed on how the carbons on the amino group of the amino acids is degraded towards to the urea, okay, and how the process of transamination and oxidative deamination is involved in the process of degradation of these amino acids. Okay, so let's take a look on the some learning outcomes that you basically were expected to get from this topic. So So from this one, it's hoped that you will get or you'll be able to relate the process that involves in catabolism and anabolisms of amino acids, and describe how essential and non-essential amino acids are synthesized, describe the function of the amino acids, and able to explain the importance of amino acids in nitrogen disposal and exclusion. Okay, so this topic will be covered on 4 subtopics, which is the first thing we're going to look at the biosynthesis of essential amino acids, okay, and then biosynthesis of non-essential amino acids, and essential coenzymes, function of amino acids, and the formation of nitrogen from the amino acids. And biosynthesis of essential amino acids, okay, so we look at the first part, which is the introduction about the metabolisms, how the catabolism and anabolisms is related to each other. And previously, we have looked at on how the catabolism process, where it's involved the large molecules, is broken into a smaller ones. Where, in this case, it will involve the releasing of the energy. So, it needs it. So in the process, it will produce the ATP, which is a form of the energy. So right now, I would like to ask you, why do you think that during the catabolism process, it's released the energy? From what you have learned before in the catabolisms of amino acids, for example, why do you think that this process released the energy? Come on, class, we have learned about this, and I would like to ask you about your opinion on this. Why is it catabolism releasing energy?
Because from one big molecule it becomes smaller molecule.
Can you relate to the process? What happened during the degradation itself? If you see from the starting of the amino acids, okay, from the starting of the degradation of amino acid until the end, okay, what makes you think that this process actually release the energy? Break down bonds, okay. Exogenic. Which process is that? They produce the energy. Which stage? Okay. If we look at the from the amino acids catabolisms itself, not considering the other part, which is lipid or carbohydrate, we just look at the anabolisms, catabolism of amino acids. Just only amino acid that you have learned before. So, at which stage actually these amino acids, the degradation of this amino acid produce the energy? Okay, redox reaction. Okay, Fatia, so which step is it? Redox reaction.
The use of energy like NADP plus H plus or NADPH become NADP, something like that.
Okay, so which stage is it? Which cycle? Okay, TCA cycle, glycolysis, citric acid cycle. Okay, so I want you to to think about it. Okay, so when we mention about the catabolisms and it's releasing energy, so I want you to know, which step in case of amino acids, it's actually produced this energy. Okay, so please relate this to all the process. Okay, from the beginning, we learn about the transformation until it enters the TCA cycle, and then and what happened to the intermediates or the side products in the TCA cycle. So so it will go to the oxidative phosphorylation, right? Yeah, in the ETC, electron transport chain. So there the NADH, NADPH, so it will be oxidized into the and release the energy, or ATP. Okay, so that is the process. So please take a look again how the process, until and try to relate to this energy releasing out of this process of catabolisms. All right, and we have another one, which is the anabolisms, which is it starts with the small molecules, but then they will be assembled and to produce the larger ones. And in this case, in the process, it will use up the energy. So it's against the catabolism, which is releasing the energy. But in anabolism, it's used the energy. And so to make the process or the metabolisms go through functions well, so we have to make sure that it's two process, catabolisms, anabolisms, are balanced. Okay, and this will make up a perfect metabolisms, which involve the biosynthesis and degradation, right? And so when we take a look at this, the characteristic of catabolism process, so we know that the process is destructive process, where it involves starting with the it started with the complex molecule, and we get a simple molecules out of this process, and it releases energy, which is catalyzed by enzymes, and is also a form of exogenous reactions. And during the process, it's involved or utilized oxygen. Yeah. So in case of anabolisms, it's a constructive process, which we start with simple molecules, and we get the complex molecule out of it, and it requires the energy where it can be stored in the form of the ATP, and it's a form of endogenous reactions. Yeah. So, and it does not utilize the oxygen. And I think I've shown you these figures before on this integration, how we integrate between the process of catabolism and anabolisms, and in between these two, we have the product with the form of the energy, in the form of the ATP, and we have this all this carrier that will be oxidized to form the energy, and we have this NAD plus, NADP, and FAD in between this process, where it will be it is ready to be oxidized, to produce the chemical energy. So this is how the integrations between the catabolisms and anabolism process it's linked to each other, towards the formation of larger or simple molecules. And this is the outline of amino acid catabolisms that we have learned in previous topic, where in case of the amino acid catabolisms, we have to remove the part of alpha amino group, and we also have the carbon skeletons where it will be degraded into another intermediate. And it depends on what types of the amino acids is it, right? So whether it is a ketogenic, glucogenic, whether it's strictly ketogenic, so all those will be degraded into different fashion, okay, different pathway, and this intermediates then it will be used or enter into another metabolic pathway where we have lipid or glucose, where it will go through this oxidation to produce the energy. And this is the intermediates that serve as the precursors and how it relates to these amino acids. Okay, so previously we have learned on how these amino acids how they are degraded into alpha-ketoglutarate or succinyl-CoA or to fumarate, oxaloacetate, or pyruvate, or to even into the acetate. And yeah, so these are divided into two forms, okay, two categories, which is glucogenic and ketogenic amino acids. All right, let's take a look at this overview of the amino acid biosynthesis. We have in case of the biosynthesis of the amino acids, of course, it's involves a different pathway than catabolism's process. And let's take a look at what are the cycles that involved during the biosynthesis of amino acids. So we started with the glucose. So let's say in the process we have the glucose and on. So here we have this ribose 5-phosphate and erythrose 4-phosphate. So this two is actually the part of pentose phosphate pathway. Okay, the purple. So this is actually these two is actually purple color sebenarnya, purple color. So these two actually relate to the pentose phosphate pathway, and these two here, oxaloacetate and alpha-ketoglutarate here. So it involves in the process of the citric acid cycles. And we also have the 3-phosphoglycerate, phosphopyruvate, and pyruvate. So these three intermediates involved in the glycolysis pathway. So these are the intermediates that involve for the amino acid biosynthesis. Okay, and so we have 20 common amino acids. 20 common amino acids. So starting with the simplest one, which is the alanine, and we have different types of amino acids, different properties, different characteristics. So it's based on the what are the side chains. Okay, side chains that actually determine the properties of the characteristics of these amino acids. So it's really good if you can memorize all this. You memorize, and actually you should memorize this structure formula for each amino acid, and you should know how to actually build the connections between these amino acids to the next one. So let's say we have peptides of these amino acids. Hopefully that you should be able to connect, understand how to connect between these amino acids, and to form a long peptides. Yeah. All right. So in case of these amino acids, we have we can categorize these amino acids in terms of whether it is non-essential or essential amino acids. So what is what can I understand from this essential and non-essential, and also conditionally non-essential and non-essential amino acids? What do you think about this category of amino acids? What is the essential amino acids?
For essential is the amino acid that our body cannot be synthesized and only can be obtained from diet. And while for non-essential is amino acid that our body can synthesize.
All right. Yeah. So what about conditional non-essential?
This one maybe sometimes our body can synthesize and sometimes it needs to take from diet. This one I'm not really sure.
Conditionally non-essential. Okay. Siapa jawab tadi? Tak apa pula your name.
Adila.
Adila. Okay. Okay, it's correct about your answer on the essential, non-essential. And about the conditionally non-essential, you said that it depends on the in the body, in the body needs, right?
Yeah.
Can you give an example? What condition is that?
This one actually I just how to say like the yeah.
All right.
Sure what
Okay.
is the condition.
Okay, Dila. Okay, thank you. How about others? Wang. Okay, Wang, where are you?
Yes.
Okay, Wang. Okay, what's your opinion about this conditionally non-essential amino acids?
It is usually not essential, but during illness and stress, then you become essential.
Okay. So meaning that during those time, we need the sources of these sources from the diet itself, right? So meaning that our body cannot produce it every time, or our body cannot produce enough during a certain condition, and we have to get it from the diet, right? So okay, so thank you. Okay, so the next one, so this one we have discussed. So, okay, so the key concepts, the key concept of amino acid biosynthesis. So like, because for this amino acid biosynthesis, we will look at the process of biosynthesis amino acids that involved in the plants and bacteria, and also in organisms, and we're going to look at the similarity of the pathway that involved in mostly in the organisms. Yeah, so in plants and bacteria, usually basically they can synthesize all the 20 amino acids, but for humans we can only synthesize about half of the 20 amino acids because we don't have the such precursors or certain enzymes that can produce all these amino acids, and all amino acids actually derived from the intermediates. Is as we have discussed on the previous topic, which is catabolisms. So we know that the amino acids is going to be degraded into the intermediates. So the same thing goes to the when this amino acid is going to be synthesized again. So we need to start with the intermediates as well. And so for biosynthetic pathway for several amino acids, such as the aromatic amino acids, it's much more complex because they involve the formation of the ring and the some chemical arrangements that involve to form the structure of these amino acids, and these are the common metabolic intermediates or pathway that involve to the formation of the amino acids. So we have the glycolysis, glycolysis, amino acid cycles, and pentose phosphate pathway. And so these three, they are actually interrelated, interconnected, link each other to provide the intermediates and the amino acid biosynthesis will go through this pathway. They involve these three pathways. And some of the amino acids, they might involve in the citric acid cycle. Some of it, they might involve glycolysis. Some might contribute to two or to one or more of this pathway, and perhaps just involve the pentose phosphate pathway. So meaning that every single amino acids, they have their own pathway to be synthesized. Okay, and as we learned in the previous topic, which is in the previous topic, we know that the amino group from the amino acid, it will be degraded into the ammonia. And then which comes from the glutamate, okay, because of the transamination. So every single amino acid becomes the glutamate, and from there go through the oxidative phosphorylation, and we get these amino group, right? So so in this case, when the amino acids is going to be synthesized, again, we need to go back to the glutamate. Okay, we have to apply, we have to ask, we have to get the demands back from the glutamate and also glutamine. Yes, so these two guys, they will provide the source of nitrogen to this formation of the amino group for these amino acids. All right. All right, so I think maybe it's 1 o'clock already, almost 1 hour. Okay, so we are going to continue on the next class lah. All right, so for this topic, and I've actually, I think I've uploaded these notes and also the next topic, which is on the amino acids. Apa yang satu lagi topik? But I think I've uploaded 2 notes in the Putra Blast, so please refer to the Putra Blast. Okay. Mana? Right. Okay, saya nak ambil attendance. Okay. Can I mention your name and just, speak up if I mention your name, huh? Okay, Shafika, are you there?
Yes, doctor.
All right. Husna,
Yes, doctor.
Sachini. Tak ada. Vartini. Kishan.
Yes, doctor.
Shin
Yes, doctor.
Fang Lin.
Yes, doctor.
Okay, Jingshi. Jingshi or Jingshi? Jingshi. Okay. Sorry. Divyasri.
Yes, doctor.
Balkis.
Yes, doctor.
Yalini. Yalini. Tadi ada kan? Mariam.
Yes, doctor.
Adila.
Yes, doctor.
Aisyah.
Yes, doctor.
Okay, those yang saya dah sebut nama, you can leave the session lah. Okay. Siapa? Aisyah? Siti Aisyah.
Doctor.
Lia.
Yes, doctor.
Okay, Nurin. Liana.
Yes, doctor.
Aini.
Yes, doctor.
Wang Meiki. Angeline. Aslam. Hanis.
Yes, doctor.
Husna. Muhammad Noor. Danuska.
Yes, doctor.
Nur Afri.